Which of the following is untrue about SAGE Genie? A. It is an NCBI web-based program. B. It allows matching of experimentally obtained SAGE tags to known genes. C. It provides an interface for visualizing human gene expression. D. It doesn't filter out linker sequences from experimentally obtained SAGE tags. The correct answer is, it doesn't filter out linker sequences from experimentally obtained SAGE tags. Which of the following is untrue about the drawbacks of SAGE? A. 1 or 2 sequencing errors in the tag sequence can lead to ambiguous or erroneous tag identification. B. Correctly sequenced SAGE tags sometimes may correspond to several genes or no gene at all. C. Correctly sequenced SAGE tag always corresponds to several genes. D. The drawback with this approach is the sensitivity to sequencing errors. The correct answer is, correctly sequenced SAGE tag always corresponds to several genes. Which of the following is untrue about SAGE? A. This approach is much more efficient than the EST analysis. B. This approach is quite less efficient than the EST analysis. C. It uses a short nucleotide tag to define a gene transcript. D. It allows sequencing of multiple tags in a single clone. The correct answer is, this approach is quite less efficient than the EST analysis. SAGEmap is a SAGE database created by NCBI. A. True. B. False. C. D. The correct answer is, true. The PCR amplification step involved in the SAGE procedure means that it requires a large quantity of sample mRNA. A. True. B. False. C. D. The correct answer is, true. DNA microarrays measure absolute mRNA expression levels without arbitrary reference standards, whereas SAGE indicates the relative expression levels. A. True. B. False. C. D. The correct answer is, false. SAGExProfiler doesn't provide information about overexpressed or silenced genes. A. True. B. False. C. D. The correct answer is, false.